Hello, my name is Richard Ray Martins and I'm head of the Molecular Neurodegeneration Research Group here in the Department of Physiology, Anatomy and Genetics. Our group is working on the molecular mechanisms of neurodegenerative diseases such as Parkinson's disease, Alzheimer's disease and the rarer disorder Friedreich's ataxia. Understanding the molecular basis of these diseases is critically important as the population ages, the number of people in the United Kingdom with Alzheimer's, Parkinson's and similar disorders will increase greatly. We need to understand the mechanisms now to develop new drugs and targets in the future. So, come on in and find out what we do. To find a cure for neurodegenerative diseases, such as Parkinson's and Alzheimer's, we first need to know what goes wrong in the brain. The brain is a very difficult place to access, and patients cannot donate brain cells or neurons. Instead, people affected by Parkinson's have donated skin cells, which are converted in a laboratory into stem cells. Once we've got stem cells, we can make any cell type we want. In our case, neurons, which make the chemical dopamine, the cell type specifically lost in Parkinson's, as we can see here under a microscope. The main task of a nerve cell is to make electrical signals to send information all around the brain and across our body. When this process is disturbed, the nerve can no longer function. Using electrophysiology, we can measure the electrical activity of neurons made from patients to understand how disease affects their function. We know that many different compartments inside a cell go wrong in Parkinson's. For example, the mitochondria make energy, like a power plant inside all cells. In the lab, we're able to test the different components of the mitochondrial power generator to see which part is broken and needs fixing. The recent increased knowledge of human genetics has greatly increased our understanding of the genetic contribution to diseases of the brain. By comparing which genes are working in a patient neuron and a healthy neuron, we can find new drug targets. If we could use a drug to reverse that change, we may even have a therapy. In Parkinson's and Alzheimer's, the protein recycling centres in a cell, labelled here with fluorescent markers, fail and cells instead accumulate gluggy protein aggregates and die. We can model this process to understand how we might stop it. Preventing or reversing this aggregation may ultimately prove to be therapeutic. When we know the cellular mechanisms behind a disease, we try to treat patient cells to improve their function. We can use state-of-the-art equipment to automate this process and screen thousands of compounds that might potentially become new drugs. We're able to use acoustics, short, sharp pulses of sound, to dispense a millionth of a milliliter of these potential drugs into plates of patient neurons. We incubate the cells with the drugs and then read out the effect of the drug on a plate reader or a high-throughput microscope, which allows us to screen for new compounds which correct faulty pathways in patient cells. Our laboratory also studies the rare childhood disease Friedreich's ataxia. This is a genetic disease in which neurons and heart cells die due to the lack of a protein called frataxin. Some patients have kindly donated blood samples, which we use to study how and why this protein is lost and test for new drugs to boost frataxin levels. Once we have identified an efficient drug through screening, we analyse its mode of action in patient cells. Once a new compound appears to be a potential therapy in patient cells for any of the disorders we study, we next test the new drug in strains of mice or rats which have the disease we want to treat before moving to humans. We can analyse brain slices to look for a reduction of disease pathology after treatment with new and untested compounds. Working with colleagues across the laboratory and the clinic, we have created a circle of translational research in Parkinson's, starting with patient samples, moving through the laboratory and ultimately working to develop better treatments and cures. Working with patients is immensely important to what we do, and we're grateful for those who've contributed samples and their time in our work. With ongoing help from these funders listed here, we're hopeful that we can further push forward our research and develop treatments ultimately for these currently incurable diseases.